Hello everyone, this is Dr. Padharia from dressmd.com. Today I am here to discuss another important topic, Lac Operon. There are many questions in the UWorld and AMBOSS guys, and please try to watch the entire video. I try to make it as simple as I can, and if you watch the whole video, I'm sure you can answer almost any question related to this topic. Okay, so let's understand what is Lac Operon and what is the gene which has, what is the function, and how it plays an important role in the metabolism of E. coli. Okay, so Lac Operon are nothing but they are the set of genes that are specific for metabolism of lactose. And what is lactose? It is a glucose. So when E. coli are growing in a media, so they need energy, and where they get the energy from? They get the energy from the lactose. And what is the function of Lac Operon in this lactose and E. coli? The Lac Operon plays a catabolic activity or breaking down the lactose so that the E. coli can get their energy. Okay, so as you can see, the Lac Operon gene, this is the DNA, they have a regulatory gene and they have a structural gene. Now, what is the function of the regulatory gene and what does it contain? So, it has a promoter site and it has a LAC-I. The structural genes, they have the LAC-Z, LAC-Y and LAC-A. Okay, and here on the next image, I'll be showing area here. This is an area for the catabolic activator binding site. This is again a promoter site and an operator site. So RNA polymerase, they come and bind to the promoter site so that there will be initiation of the transcription. Now, what is the function of a regulatory gene? The regulatory gene, which has the LAC-I, it produces a protein called the repressor protein. Now, how does the LAC-I produces a repressor protein? LAC-I from the DNA, when it produces RNA and mRNA with the transcription, that's the transcription, that's the definition of a transcription. And when the RNA and the mRNA, when it produces a repressor protein, that's called the translation. So guys, you should remember that what is the difference between a transcription and translation. Transcription means when RNA or mRNA is formed from the DNA, and translation means when a protein is formed from the RNA. So what is the function of the repressor protein? This repressor protein, which is produced by the LAC-I of the regulatory gene, it can go and bind to the operator site, inhibiting the transcription. Okay, so that is why the regulatory gene is regulating the structural gene. As a result, it is called regulatory gene. Now, what is the function of a structural genes? The structural genes function is to produce enzymes, and enzymes are nothing but proteins. Okay, so we can see that here that the structural genes like LAC-Z, it produces beta-galactosidase, LAC-Y produces permease, and LAC-A produces trans-acetylase. Now, what is the function and how they are produced? As you can see, this DNA in the structural genes, this DNA ultimately with the transcription they are forming RNA, and with the translation they are producing protein or the enzymes. So the function of these enzymes, guys, like I said, LAC-Z produces beta-galactosidase that ultimately forms lactose, which converts into glucose and galactose. Lactose also produces enzyme called the allolactose, okay, so which binds to the repressor protein. And what is the importance of allolactose binding to the repressor protein? What is the advantage? I'll be telling in the next image. Okay, so we can see here LAC-Z produces beta-galactosidase, LAC-Y produces permease. What is the function of the permease? So what happens when the E. coli is growing in a media, any media that helps in the uptake of the lactose, and that is the function of the permease. So another enzyme that is produced by the LAC-A. So LAC-A produces trans-acetylase, and this trans-acetylase, they are they produces an acetyl group. Okay, so what is the advantage of this trans acetylase producing an acetyl group? Because during when the E. coli are growing in a media, they produces what is called the thiogalactosides. What are the function of these thiogalactosides? They are toxic to the growth of the E. coli. So these trans acetylase, they will produce an acetyl group that will make it non-toxic, so the bacteria can grow. Again, the trans acetylase is formed by the Lac A, and they produce acetyl group. When the E. coli is growing in a media, they produces thiogalactosidase that are toxic for the growth of the E. coli. So when the trans acetylase, they produce the acetyl group, they make this thiogalactosidase non-toxic. Okay, so acetyl group will make it non-toxic, so the E. coli can grow. So guys, in this image we learned that what is a regulatory gene, what is structural gene, what are the enzymes it produces, and how does the regulatory gene regulates the structural gene, and then what are the promoter and the operator side. And then in the next image we will be discussing the relation of catabolic activity and the glucose. So you can see this beautiful image, guys, that for a Lac Operon to be strongly expressed, okay, they should have low glucose and there should be presence of lactose. Lactose should be available and glucose should be absent for the Lac gene to be strongly expressed. So now how will you explain this concept? So as you can see, when there is a low glucose in the media, they can activate an enzyme called adenyl cyclase. What is the function of adenyl cyclase? It converts the ATP to cyclic AMP, and that will stimulate the catabolic activator protein, and that will go and bind to this catabolic activator protein site, which is just before the promoter site. Okay, like we saw in the previous page, we saw the promoter and the operator, and before that we have the cap site. So again guys, low glucose will activate the adenyl cyclase enzyme that will convert the ATP to cyclic AMP, and cyclic AMP will stimulate catabolic activator protein, and that will go and bind to the catabolic activator protein site, and that will induce this transcription. That means it will stimulate the production of transcription will occur, and then translation, and then proteins will form. So this operator is active. Why the operator is active? Because lactose is available. So if I explain that concept here, let's say as we saw in the previous page that the lactose, it produces an enzyme called the allolactose. Now what is the function of allolactose? These allolactose, as we saw, the Lac A is producing a protein called the repressor protein. So if we remember the concept, guys, like a mother and a baby, let's say there is a mother, okay, the mother is nothing but allolactose, okay, and it is holding the baby, that is repressor protein, okay, as you can see in the previous image. So if the allolactose is the mother, and then allolactose is coming from the lactose, right? Lactose is coming from the beta galactosidase. So these allolactose will go and hold this repressor protein. So when it holds this repressor protein, this allolactose, this repressor protein cannot go and bind to the operator site. So the operator site is active. So now you understand, guys, that why lactose is good, because lactose will produce allolactose. Allolactose is the mother holding the baby, that is the repressor protein, so that the baby cannot go here and there. And then the operator is active, and then when there is low glucose, the cap is getting activated, the promoter is getting activated, the RNA polymerase will come and bind to the promoter, the operator gets activated, and the structural genes are activated to produce the matured protein. Okay, so the lac genes are strongly expressed in a situation where there is a low glucose and lactose available. This is one scenario, guys. What will happen? Can anyone tell me in the comment box that what will happen when there is a high glucose and lactose is not available? Of course, lac genes will not express. So how will you understand that concept? So if we understand that concept here, that this we understood it very well. Now, if we understand this one, let's save this. B, okay, so high glucose and lactose is not available. So what will happen, guys? As we said, if there is a high glucose, the high glucose will not stimulate the adenyl cyclase, because low glucose will stimulate the adenyl cyclase. So no cap is getting activated. So when no cap is getting activated, there will not be any transcription. Similarly, we said lactose is not available. Yes, when mother is not there, when allolactose is mother is not there, the mother cannot hold the baby, that is the repressor protein. So the baby can move here and there and sit or here and there and sitting on the operator. So when it sits on the operator, what will happen? The operator is not active now and the lac genes are not expressed. So you can understand that high glucose is not good, cap is not getting activated, and if glucose is not there, then the allolactose cannot inhibit the repressor protein, and the repressor protein will go and bind to the operator side, and lac genes are not expressed. Okay, I hope that is clear. The third scenario, guys, is a low glucose and lactose are unavailable. Yes, lac genes are not expressed again. You see, what will happen if there is a low glucose? Yes, low glucose is there. So that is activating the adenyl cyclase and activating the cap and the cap binding side. So low glucose is good, we understand that, but lactose is not there, lactose is not available. So when that lactose is not available, it is not able to bind to the repressor protein. So the repressor protein is free and going and binding to the operator site like this. It is just like locking the operator site like this. Okay. So you can see this is a repressor protein binding to the operator site, because there is no lactose is not available. You see that there is no lactose available. So the repressor protein can go and bind to the operator sites and the lac genes are not expressed. Okay. So what will happen when there is a high glucose and lactose is available? Okay. So high glucose, yes, high glucose again, the adenyl cyclase is not activated and cap is not activated, and lactose is available. So when lactose is available, what will happen? Yes, this lactose will inhibit the repressor, bind to the or hold the repressor protein, so that the repressor protein cannot go and lock this operator site. So this operator site is active because lactose is there to hold the protein, repressor protein. So it is not able to bind to the operator site. So the operator is active, promoter is active, but cap is not active. Why cap is not active, guys? Because there is a high glucose. They can only be activated with the low glucose. So lac genes are expressed, but they are expressed very low, or the basal expression. Okay, this is how, guys, the E. coli, they utilize lactose, and the Lac Operon breaks the lactose and they use as an energy source. And what happens in a scenario where there is a low glucose and high glucose and lactose available and lactose not available, these are the scenarios they may give you. They may ask about a research scientist is doing this, blah blah, and then they will give a different scenario. It's not tough at all, guys. If you try to understand the image which I explained you, try to watch two to three times, and I'm sure you'll get it. I try to explain very easily, and my students are watching after watching this video, or after my students after the session, they are getting the answers correct. Okay, so try to watch them, try to do the question, and please let me know in the comment box, guys, did it help you or not. Okay, and please visit my website dressmd.com. You guys take care and have a great day.